Hi, welcome back to chapter 14. This week we will be looking at how proteins are made. So we'll be talking about genetic transcription, translation, and regulation, essentially how the DNA code is read and proteins are made from that DNA code. So 14.1 looks at the structure of proteins. So we'll talk about first what proteins are made up of and how they get made from the DNA code as an overview. So on the top of this picture here, you see the DNA code. Remember, it's a string of what we call nucleotide bases all strung together, and they're sort of in this double-sided spiraling shape. And they call this a double helix because there's two sides to it, and helix is sort of referring to the spiral shape. So the DNA code gets read 3 bases at a time. So if we're to look at our DNA code here, we see GCA is the first 3 bases, GCA. And then next we have AGA, AGA, and so on. Now, the code gets read 3 bases at a time, and each set of 3 bases produces one of what we call an amino acid. So these 3 bases produce the amino acid alanine. These 3 bases produce the amino acid arginine, and so on. When a segment of DNA is read, one amino acid is produced for each series of 3 bases that's read. So if you look at this chart here, it shows all the different possible 3-letter combinations that you can have, 3 letters of DNA bases here in black. And then the colors on the side here show what amino acid is made from those 3 bases. This is a chart here showing the 3-letter code and the name of the amino acid. So if you were in your DNA code, you had the bases UUU or UUC, you would produce the amino acid PHE, which as we can see from our code, PHE stands for phenylalanine. So when you have UUU or UUC, you get phenylalanine. If you had UUA or UUG, you would get the amino acid leucine. So LEU stands for leucine, and so on. Now there are 20 different amino acids that can be produced from this code here. Some amino acids code, some letters, series of 3 bases code for amino acids, and some 3-letter codes are stop codons, and that tells your body to stop reading the DNA code. The code is done. So you can either have a stop codon, a start codon, or an amino acid produced from a codon. So here are the 20 different amino acids. This is the same as the chart that we just saw, but essentially our DNA can code for 20 different amino acids. So these are the 20 amino acids here that your body can produce. These 20 different amino acids can be arranged in different orders to make all of the different proteins in our body. So again, amino acids are given 3-letter abbreviations. A string of amino acids is called a polypeptide. So when you start stringing as your code gets read, your DNA code gets read, the amino acids start getting put together in a chain. And this chain of amino acids is called a polypeptide. So this chain of amino acids is going to make up the polypeptide that will make the protein insulin for this picture here. Now a polypeptide is a chain of amino acids. It doesn't become a protein until it's folded. And so the folded structure of this chain is important to the functioning of the protein. So this here is a chain of amino acids or a polypeptide. This here is the protein because it is folded and ready to be used in the body. Once a polypeptide chain has been folded into its working 3-dimensional shape, it's called a protein. Although there are hundreds of thousands of different proteins, all of them are put together from a starting set of 20 different amino acids. So those amino acids can be put together in different lengths and different orders. Some proteins will have some of those amino acids. Different proteins will have different combinations of those amino acids. So not every protein has every amino acid, but all proteins are made from a combination of one or more of these amino acids. It's the order in which the amino acids are linked in that polypeptide chain that determines what protein will be produced. And proteins are often composed of 2 or more linked polypeptide chains. So in that previous slide, we showed a picture of what the protein insulin looks like. That was one polypeptide chain, but most proteins are going to be composed of 2 or more polypeptide chains that get made and linked together as they get folded up. Next, let's look at section 14.2, protein synthesis in an overview. So first, there are 2 stages of protein synthesis. The 2 stages are what we call transcription and translation. So first off is transcription. This is where the DNA code gets read. A copy of that DNA code gets made. This is something called mRNA or messenger RNA, and the messenger RNA leaves the nucleus. So that's the first stage of your DNA getting read, is that a copy is made and the copy leaves the nucleus. Your original DNA never leaves the nucleus. You need that DNA code to always stay inside your nucleus so it can continue telling your cell what to do. But this copy gets made, these copies get made constantly, and they leave the nucleus and move to a ribosome. So this mRNA here gets created, that copy of the DNA goes to the ribosome, and in the next stage of protein synthesis, we call this translation. This is where something called transfer RNA reads the DNA code 3 pieces at a time. So these tRNA pieces come, they read the DNA code 3 pieces at a time. They go out and they collect the amino acid that segment of 3 DNA bases codes for. They collect the amino acid, they bring it back to the ribosome, and they start connecting those amino acids together in the order specified by the messenger RNA. And remember, messenger RNA is just a copy of your DNA. So this is where the DNA code gets read and where the amino acids that it codes for get put together in the appropriate order. So first up, let's look at the first step of protein synthesis, which is transcription. So remember that there are 2 stages to protein synthesis. And again, protein synthesis is where we read the DNA code gets copied and gets taken out of the nucleus and made into a protein. So there's 2 stages for that, transcription and translation. And let's look at transcription first. So essentially transcription is when you take the DNA code and you make a copy of it. And that copy is what's going to leave the nucleus and go out and complete the steps towards making a protein. So you have your DNA code here, which is 2 strands, and a single strand copy gets made of that DNA. That single strand is called RNA, and we'll go over in the next slide how exactly that happens. Okay, so our first step of transcription starts with the DNA code getting unwound. So our DNA chain here is hooked together. There's 2 chains. They're hooked together in the center, these bases, by hydrogen bonds. So this enzyme called RNA polymerase unwinds the DNA 1 segment at a time. So it'll start here and it'll just continue to slowly unwind down the DNA chain. And what happens then is new bases are added to one side of this chain of something called RNA, which is very similar to DNA, except RNA is just a single strand. And so essentially a base will start getting added onto one side of the DNA. So wherever there is a G on the DNA, a C will get added. Remember, G and C are complementary to each other. So if there's a guanine, a cytosine will get added. If there's a cytosine, a guanine will get added. So it's similar pairing as you would see in DNA. The only difference is in RNA, there is no T in the RNA code, and instead there is a U. So in DNA, A and T pair up together. In RNA, A and U pair up together. So if there is a T, an A will still be added, but if there's an A U will be added. So enzymes just come along and they start adding all these bases onto the DNA code wherever it's unwound. And again, it's just a single strand, and in that strand, after it's made, will come unhooked from the DNA, and that means you have a copy then of that DNA code. And this making a copy of the DNA code is called transcription. So next up, let's look at the second step of protein synthesis. We've made the copy of the DNA code in step 1 in transcription. So now we have step 2, which is translation. If you remember from Bio 101, ribosomes are little organelles that are floating around in the cell's cytoplasm. So they're little tiny dots that, if you remember from looking at the cell, and they are where proteins get made. The way that this happens is we have a ribosome here, and then we have our messenger RNA code that got copied from the DNA. So it's the single-stranded code of what we now call RNA instead of DNA, and the code starts getting read 3 segments at a time. So if there is a UUU in the code, then these pieces here, which are called transfer RNA or tRNA, they're just floating around in the cell, they will go out and they will collect the right amino acid that corresponds to this code. So if you have UUU on your code, UUU, that transfer RNA will go out and collect the amino acid phenylalanine or PHE, and it will bring it back to the ribosome. Then it will read the next segment of the code. Let's say it's CUU. So CUU corresponds to the amino acid lutein, and the transfer RNA will go out and collect this LEU, lutein amino acid, and bring it to the ribosome. And so a chain of amino acids starts getting formed by reading this code. Let's review those steps again. The RNA code gets read, this code down here. The tRNA brings the corresponding amino acid to the mRNA strand, so it brings in the corresponding amino acid. The amino acids get strung together in step 3, and this string of amino acids is called a polypeptide chain. Remember, it's not a protein until it gets folded up. So at this point, it's essentially a protein, but not folded into the correct shape yet, and we call that a polypeptide chain. Next up, let's look at section 14.5 on genetic regulation. We've talked about how DNA gets read and how proteins get made, but it's important to know that protein production is carefully controlled or regulated in living things. And what this means is that proteins are not constantly being made in your body. DNA is transcribed, and proteins are made in accordance to the needs of an organism. And less than 2% of the DNA in the human genome actually codes for proteins. There's a lot of other DNA that serves other purposes. Some sections of DNA just may be things that don't code for anything at all, but a lot of the segments that don't code for proteins make an organism more complex, and we'll get into that coming up, how that works. But other segments of the DNA code are regulatory, meaning they help regulate the production of protein. So they help the body know when it needs to produce a protein. So they don't actually produce a protein themselves. They help tell the body when to make proteins. Now, transcription requires that RNA polymerase be properly aligned at a sequence of DNA bases called a promoter that lies just upstream from a DNA sequence. So there is something called the RNA polymerase, if you remember, starts unwinding the DNA. So it has to hook on to this promoter area in order to start transcribing the DNA and making proteins. So this promoter is sort of the segment of DNA that says, start making DNA here. There's usually a second non-coding segment of DNA called an enhancer. So this doesn't code anything, but it is an enhancer, and it lies some distance from the promoter sequence. So somewhere before this promoter is something called an enhancer. And as you can see in this picture here, if there's a better alignment of the enhancer proteins, you will transcribe at a higher rate. So you will transcribe more of that DNA segment. So it's the same DNA segment here, but when your enhancer is better aligned, it determines how much of that, how often that DNA code gets read. And so, for example, in a chicken, you have not as great of alignment of the enhancer proteins. You can see these side pieces here are not aligned as well. In the mouse, the enhancer proteins are aligned a little bit better. This segment of DNA that codes for the production of the thoracic vertebrae, when it's better aligned, more thoracic vertebrae will get made. In the chicken, it's not as well aligned, and less thoracic vertebrae will be made. So you could have the same segment of DNA, but just a different alignment of an enhancer protein on that DNA and get a completely different, not different structure, but same structure, but more of that structure made based on the alignment of that enhancer protein. So let's look now at those extra pieces of DNA, as we mentioned earlier, that are non-coding pieces of DNA. So the initial RNA chain that's produced during transcription, which is called the primary transcript, undergoes editing by means of some sequences called introns being cut out of it. So the introns you can see here are these in purple. And so the remaining sequence, the exons, are spliced back together. So your DNA, this initial DNA code or RNA code, sorry, gets processed, and then these introns are cut out. So they kind of fold up and are removed, and what you're left with is the remaining segments of the RNA code, and this completed section is the messenger RNA section. So introns don't code for proteins, but most exons do code for proteins. So again, part of your DNA code is used to make proteins, and part of that DNA code gets removed, and we'll talk about that continuing on the next slide. So some relatively simple organisms have nearly as many genes as human beings do. So if you look at the Arabidopsis thaliana, the mustard plant, it has 25,000 genes. That's about what the human has. But human beings are able to be much more complex than this organism, thanks in part to a form of genetic regulation called alternative splicing, in which the primary transcript can be edited in different ways. So if you remember, we looked at that RNA code on the last slide, and certain introns were cut out and certain exons were left. Now, different introns and different exons can be removed and left depending on what protein needs to be made, so you can cut out different segments. Now, a more simpler organism tends to have less sort of extra DNA, and so it's read straight through with not as much being cut out of it. Humans, we have a lot of genes that aren't read all at one time, but that DNA might be read at a different time. So here's an example. We have this primary transcript that got made from the DNA. This example on the top. So here we have introns and exons. So in the example on the top, exon 1, 2, and 4 are spliced out and put together, and it makes protein A. In another example, the code might get read and exons 1, 3, and 5 might be used and spliced back together, and then you would get protein B. So you can keep different combinations of introns and exons and make different proteins based on those different segments. So what's important to remember here is we have a DNA code, and our DNA code codes for different things, but that code can be read differently at different times and put together in different pieces to make different proteins. So the same segment of DNA does not always code for the same protein. Now, segments 1, 2, and 4 would always code for this protein. Segments 1, 3, and 5 would always code for this protein, but depending on the needs of the organism, they can select different exons and put them together and make different proteins. So DNA codes for several different forms of RNA. Of these, only messenger RNA then goes on to make proteins. DNA also codes for several varieties of regulatory RNA that undergo the collective name microRNAs. Of the 1,500 microRNA sequences discovered to date, all have the effect of reducing the production of particular proteins, usually by targeting their messenger RNAs for destruction. So you have all these little segments of RNA, and these little segments are called microRNAs, and they in effect regulate when a protein will be made. So they destroy certain segments of the mRNA, this code made from your DNA, they destroy certain segments of it and say, so essentially that segment's not going to make a protein at that point. So this is, again, these are pieces of your DNA that are not used to make protein, but instead are used to regulate the production of proteins. It's not genes, but the regulation of genes. It's the most important factor in bringing about the differences among organisms. So there's little relation to the number of genes an organism has and the complexity of that organism. So if you look at, again, a human and a mustard plant have almost the same number of genes. What's different between some very simple things and some very complex things is that they have a higher portion of DNA that does not code for protein. So humans have 98% of the DNA that doesn't code for the making of proteins. So most of the DNA inside a human is, again, that regulatory DNA, that DNA that's going to regulate when and where it's read. So when you can regulate and mix up how your DNA is read, you can get some really different combinations and very complex organisms. Very simple organisms, most of their DNA is going to code for a protein, and it's almost always going to be read in that same order because it's not being regulated as much. Genetic regulation and non-coding DNA may be more important to genetics than has traditionally been assumed. We have no definitive answers about these questions, however, because research on genetic regulation and non-coding DNA lies at the cutting edge of contemporary research. So we do know that non-coding DNA is very important, but it's something that we are just starting to learn about in the field of genetics. That is the end of our Chapter 14 lecture, and I will be in touch with you again next week for Chapter 15. If you have any questions as you go about your week, please let me know.